Hello friends, myself Dr.Subhash Mishra and I am a microbiologist and today I am talking about history of microbiology. So kindly watch this video and update your knowledge. What is microbiology? Study of different microorganisms. It can be bacteria, viruses, parasites, fungus. When we discuss about medical microbiology, it means study about bacteria, viruses, fungi, protozoa, and elements, and how they produce the disease inside the body, how it can be diagnosed in the laboratory, how it can be treated, what is their treatment protocol. Along with treatment, we also study about how the disease can be prevented. When we discuss about history of microbiology, a great contribution of Anthony van Leeuwenhoek, that is development of microscope. By microscope, we can easily study microbes. Next, development of smallpox vaccine is done by Edward Jenner. Louis Pasteur, a great contribution, that is sterilization, removal of harmful microbes from the article. It may be a steam sterilizer, hot air oven, and autoclave. These are different sterilization techniques which are used in different sterilization methods. When we discuss about growth needs to bacteria, as we know that in laboratory during culture, microbes require different types of medium for their growth. So these are invented by Louis Pasteur. When we discuss about anthrax, then Louis Pasteur also studied about anthrax bacillus. A very important technique, that is pasteurization. How we can preserve milk for a long time. This pasteurization technique is developed by Louis Pasteur. When we discuss about antiseptic, it is given by Joseph Lister. A very important contribution because during surgery, antiseptic have a very important role. When we discuss about Robert Koch, the founder of modern bacteriology, contribution of Robert Koch, concept of smear making and staining with aniline dyes. During bacteriological study, we make a smear in the laboratory and we stain it and understand the shape of the bacteria. That is very important technique. Number two, culturing. As we know that concept of cultivation of bacteria on solid media and hanging drop methods are discovered by Robert Koch. Robert Koch also discovered the causative agent of TB, cholera, and pink eye. Robert Koch also discovered Koch postulates. He proved how produces infection. He developed the method for testing disinfectants and he got Nobel Prize for his work in tuberculosis and he got Nobel Prize in 1905. When we discuss about Koch postulates, Koch discovered rod-shaped bacteria called bacilli in patient tissues but needed more evidence that they were the cause of disease. He formulated 4 conditions that came to be known as Koch postulates. The organism was present in every case of the disease. Number 2, it could be cultured outside the body. Number 3, inoculation of the cultured caused the disease in an animal. Number 4, the organism could be found in the animal. In short, Koch postulates says that during infection, must be a causative agent of any infections, and that causative agent can be grown in the laboratory. And when we again introduce in the healthy person, they produce the same infections, and that can be identified in the laboratory. When we discuss about Rox and Yersin, they understood the mechanism of pathogenesis. How disease produce, that is called pathogenesis. During their study of diphtheria toxin. Paul Ehrlich, a great contribution to understand about toxins and antitoxins. Ernest Ruska, the development of electron microscope. By the help of electron microscope, we can easily study internal structure of the cell. Karl Landsteiner studied about immunity, the defense power of the body against infections. They got Nobel Prize in 1930. They discovered different types of blood group. Alexander Fleming discovered antibiotic, penicillin. Carolus Linnaeus nomenclature, a very important during classification, that is taxonomy, like kingdom, phylum, class, order, family, genus, and species.
Thank you so much for watching this video.